Okay, when you have all of the counts for each individual person, you have to figure out what is significant. So one thing to take in mind is if this person has 4,985 counts of A2M and another person has, say, 27 counts of A2M, one of the first things that you should ask is, did this person with a higher number just have more reads that were sequenced? Because if you have more reads that were sequenced, maybe this gene would be elevated. But you don't want to go through and ask that question for every subject. So you want the program to do it for you. And once you get all of these raw counts, you're going to put it into something that does differential expression analysis. And differential expression analysis basically takes into account all of the questions you could ask of like, well, what if this is because of this? What if this is because of that? It's not foolproof and it's always evolving. There are tons of differential expression softwares, and which one you use and which one works is just sometimes a matter of taste. There are some really bad ones, there are some really good ones. I try and rely on studies that compare different programs, and then I also run a few different ones and kind of use my own common sense of what looks right. And that goes back to like the picture representation of genius and what I can see, as well as just some basic numbers I run. Anyways, you won't have to figure that out, but just know that the field is always evolving in the computational biology and this type of stuff. So what you use now may not be the best option in the future, but I guess all you need to know is after you have the raw counts, you do differential expression, and you'll use Galaxy for that. Probably under RNA-seq. I'll tell you exactly what to use. So feature counts is one way of getting counts. I'm using StringTie, which does assembly, but there's HTC count. This is really common in papers. Might have you use that. After you do the counts, though, you have to do differential expression, and anything with DE is differential expression. And then after so that basically compares different groups. So I have one run, I just have no clue where it is. Differential expression using Trinity assembly, that would take forever. So Trinity de novo assembly of RNA-seq data. That's if you don't have a reference genome to map to. So de novo means from scratch. And that would be just taking the FASTQ files and having an assembly occur. Very long time to run, unless you have a good supercomputer to tap into. And we don't need to do that because we're working with human. It's really if you're working with an organism that's not sequenced. Mapping is by far the best way to get your information. It's fast and easy. There's less problem. I wonder if I can do. There's less ambiguity problems. So basically, when you're running DSeq, you're going to enter in the groups. So with our guys, we have no matching results. Let me pull up the subjects sheet. And all of this, very importantly for you to know, all of this you're doing blind, which means that you don't know what BRAC score apply or Thal score applies to what person. I don't think Thal has said Thal score. We usually work with BRAC because we're a Thal lab, but copy Christie cases. So this, I work in Excel a lot. If you don't know Excel, you'll probably learn it working with me. I color code everything. So blue is low colors, yellow is high. These are all of our subjects. You don't know what any information on the subjects when you're doing anything, and that's called working blind. When I write manuscripts, I always put that analyses were done with a blind researcher or blind to the researcher, and then Elliot changes it because I think it's funny. But he'll change it to like blind to the subject information. I just put everything was done blind. I was blind. These people actually I don't know if they messed up, but everything that starts with, oh, you can't see this. Everything that starts with 1 is actually a male, and everything that starts with 2 is a female, which is really bad. So usually you would have someone recode it for you, or you would recode it yourself. I didn't realize this until too late, but what I'm actually doing, I have a recoded system, and so there's no way of telling which one is which subject. I lost my train of thought. Oh, yes, the BRAC stages. Okay, so here, or Brock, I think it said Brock. Brock. So here if I sort this, what we actually have is we have, let me see, 6 Brock 3, 12 Brock 4, 5 Brock 2, and 3 Brock 1. I thought we had more of an equal division between 3 and 4. I'll have to think about this. Anyway, because we only have 3, we probably don't want to leave that as one group, so my plan is to combine 1 and 2, keep 3 as it is, and combine 4, keep 4 as it is, so we would have 3 groups. So when you're doing the differential expression analysis, you would specify that all of these guys are in one group, all of these guys are in another group, all of these guys are in a different group. And then what the differential expression software does is it takes into account questions like, does this person just have a high number, assuming this is a high number, does this person just have a high number of A2M because they had more reads sequenced, or is it actually high expression? Differential expression software takes care of all of those questions. You have to, if you're doing this on your own, you have to research whether it takes care of that. It should take care of that. And then it also compares the groups. So it's going to combine all of these subjects and compare it to all of these subjects, and it's going to do that for all of the genes. So, nice. The software does most of the stuff for you. Then after you get that data, you start making graphs and doing statistics. I'm starting a video.